Hello everyone, welcome to this detailed video on HPLC calibration. In this video, we will understand HPLC calibration from the basic concept to practical execution. We will discuss what HPLC calibration actually means, why calibration is required, which HPLC parameter need to be calibrated, how calibration is performed, what standard and instruments are required, how to calculate the results, what are the typical acceptance criteria, how to document calibration, common calibration failures, and finally what an analyst should check before releasing an HPLC instrument for routine analysis. So let's start with the basic concept. What is HPLC? HPLC stands for High Performance Liquid Chromatography. It is one of the most commonly used analytical techniques in pharmaceutical quality control laboratories. An HPLC system separates components present in a sample and allow us to identify and quantify those components. A typical HPLC system consists of a mobile phase reservoir, degasser, pump, injector or autosampler, analytical column, detector and chromatographic data system. For reliable analytical results, every critical component of the HPLC system must perform within its specified requirements. This is where calibration becomes important. What is HPLC calibration? So what exactly is HPLC calibration? Calibration is the documented comparison of an instrument's measured performance against an established standard or reference value using a defined procedure and predefined acceptance criteria. In simple words, calibration tells us whether the instrument is giving the correct and reliable response. For example, suppose we use a calibrated volumetric device to prepare a known solution and inject it into the HPLC. If the instrument measures the expected response within the predefined acceptance criteria, the relevant parameter can be considered acceptable. The important point is that calibration is not simply checking whether the instrument is working. It is demonstrating with objective evidence that the instrument performs within predefined requirements. Calibration versus qualification versus system suitability. One common question is, what is the difference between calibration, qualification and system suitability? Calibration generally evaluates a specific measurable characteristic against a reference standard. Qualification demonstrates that equipment is appropriately installed, operates correctly and performs as intended. System suitability, on the other hand, is normally performed as part of an analytical procedure before or during routine analysis. For example, system suitability may include parameters such as theoretical plates, tailing factor, resolution, and injection repeatability or RESD. Therefore, calibration and system suitability should not be treated as the same activity. Why HPLC calibration is important? Why is HPLC calibration important? Because analytical results generated by an HPLC are used for important pharmaceutical quality decisions. Incorrect instrument performance can potentially affect assay result, impurity results, dissolution results, content uniformity results, and many other analytical activities. Proper calibration helps provide confidence that the instrument is performing consistently and within established requirements. It also supports regulatory compliance and data integrity. Important HPLC parameters. Now let's discuss the major HPLC parameters that may be evaluated during calibration. The exact parameters depend on the HPLC model, detector type, laboratory procedure, manufacturer recommendations, and applicable regulatory requirements. Common performance check includes pump flow rate, detector wavelength accuracy, injector or autosampler performance, injector precision, column oven temperature, autosampler temperature, gradient composition or proportioning, or GPV, detector response or detector linearity, pressure and leak related checks, and other manufacturer specific functions. It is very important to remember that there is no single universal HPLC calibration checklist applicable to every HPLC model. The calibration procedure should always be based on the approved SOP, manufacturer recommendation, applicable pharmacopoeial or regulatory requirements, and the laboratory qualification strategy. Flow rate calibration. Let's start with one of the most important parameters, pump flow rate. Flow rate calibration verifies whether the actual flow delivered by the pump corresponds to the programmed flow rate. For example, suppose the HPLC is programmed at 1 ml per minute, the outlet can be directed into an appropriate calibrated collection device such as volumetric flask, and the delivered volume is measured over a defined period. The measured flow rate is calculated using flow rate equals collected volume divided by collection time. For example, if 10.05 ml are collected in 10 minutes, 10.05 divided by 10 gives approximately 1.005 ml per minute. The result is then compared with the acceptance criteria defined in the approved calibration procedure. The actual procedure, collection method, stabilization time, and acceptance limits should always follow the approved SOP. Why flow rates matter? Why is flow rate so important? Because flow rate directly influences chromatographic behavior. A significant difference between programmed and actual flow can affect retention time, resolution, peak shape, pressure, and ultimately analytical performance. Therefore, accurate and consistent pump delivery is essential. Wavelength accuracy calibration. Next is the detector wavelength accuracy, particularly for UV or PDA detectors. The purpose is to verify that when the detector is programmed to a particular wavelength, the actual wavelength response is within the specified tolerance. A suitable certified reference material or reference standard with known spectral characteristics may be used according to the approved procedure, such as caffeine. The detector records the spectrum and the observed wavelength. Maximum is compared with the certified or stabilized reference value. For example, if a reference material has a known wavelength maximum, the instrument observed maximum should fall within the predefined acceptance range. The exact reference material, wavelength values, procedures, and acceptance criteria depend on the detector and the approved calibration procedure. PDA detector. In a PDA detector, we can obtain spectral information over a range of wavelengths. This makes wavelength accuracy especially important. A wavelength error can influence the measured observance and potentially affect quantitative results. Therefore, detector wavelength performance should be periodically verified using an appropriate reference. Injector precision. Another important parameter is injector precision. Injector precision evaluates whether repeated injection of the same solution produces consistent response. For example, the same standard solution can be injected multiple times according to the approved procedure. The peak areas are recorded and the percentage relative standard deviation or percent RSD is calculated. The formula is percent RSD is equal to standard deviation divided by mean multiplied by 100. The calculated percent RSD is then compared with the acceptance criteria specified in the approved procedure. A high percent RSD may indicate problem related to injection performance, sample preparation, autosampler operation, solution homogeneity, carryovers, leaks, bubble, or other chromatographic issues. Therefore, investigation should not automatically conclude that the autosampler is defective. Column oven temperature. Next is the column oven temperature accuracy. Temperature can influence retention, selectivity, viscosity, and chromatographic performance. The column oven is set to a specified temperature, allowed to equilibrate, and the actual temperature is measured using an appropriate calibrated temperature measuring device. The measured value is compared against the approved acceptance criteria. For example, if the set temperature is 30 degrees Celsius and the measured temperature is 30.2 degrees Celsius, the result is assessed against the applicable tolerance. Temperature mapping. In some calibration or qualification programs, temperature mapping may also be required. Multiple locations can be evaluated to understand temperature uniformity. The exact requirement depends on the equipment design, intended use, SOP, and qualification strategy. Again, always follow the approved procedure rather than applying an assumed universal acceptance limit. Autosampler temperature. For system equipped with temperature controlled sample compartments, autosampler temperature may also require verification. This is particularly important when the analytical procedure requires samples or standard to remain at a controlled temperature. The actual temperature is measured using a calibrated reference device and compared against the predefined requirements. Gradient proportioning or GPV. For gradient HPLC system, another important consideration is solvent proportioning or gradient composition, known as GPV. The purpose is to verify that the system delivers the intended mobile phase composition according to the programmed gradient. The exact test procedure depends heavily on the HPLC design and the approved calibration method. Some systems use low pressure mixing while others use high pressure mixing. Therefore, the calibration approach must be appropriate for the specific HPLC configuration. Detector response or linearity. Depending on the detector and laboratory procedure, detector response characteristic may also be evaluated. Known concentration can be analyzed and the detector response can be evaluated across an appropriate range. The resulting data may be assessed using a calibration curve or other predefined statistical criteria. The acceptance criteria should come from the applicable SOP or validated procedure. Pressure and leak check. Before and during calibration, the HPLC should also be checked for leaks and abnormal pressure behavior. Potential leak location include pump connections, tube fittings, injector connections, column fittings, and detector flow cells. Abnormal pressure can have multiple causes, including blocked tubing, contaminated filters, column problems, air in the system, mobile phase issue, or mechanical problems. Pressure monitoring is therefore an important part of routine HPLC maintenance and troubleshooting. Although pressure itself is not necessarily a standalone calibration parameter. Materials required. Before starting calibration, ensure that all required materials and measuring devices are available. These may include certified reference materials, calibrated temperature measuring devices, calibrated valences, timers, volumetric equipment, appropriate solvent, and the approved calibration procedure. One critical point is often overlooked. All reference and measuring devices used for calibration should have valid calibration status. Pre-calibration check. Before beginning calibration, perform a pre-calibration check. Verify that the HPLC is clean and suitable for use. Check the PM or preventive maintenance status. Verify that there are no visible leaks. Check solvent availability and suitability. Confirm detector and lamp status. Verify the status of required reference standard. Check that all measuring instruments have valid calibration status. Confirm that the correct software and approved procedure are being used. Only after this check should calibration begin. Calibration execution. The calibration should then be performed according to the approved procedure. The general sequence is: first, prepare the instrument and required materials. Second, allow the system to stabilize as specified. Third, perform the individual calibration parameter. Fourth, record the raw data. Fifth, calculate the result. Sixth, compare the result against the predefined acceptance criteria. And finally, document the complete activity. Example of calibration results. Let's look at a simple example. Suppose the program flow rate is 1 ml per minute and the observed flow rate is 1.005 ml per minute. If the result meets the predefined acceptance criteria, the test passes. Similarly, if the column oven is set to 30 degree and the measured temperature is 30.2 degree, the result is assessed against the approved tolerance. The important point is that we should never decide pass or fail based on personal assumptions. The result must be compared against the approved acceptance criteria. What if calibration fails? What should you do if an HPLC calibration test parameter fails? Do not simply repeat the test until you obtain a passing result. First, stop and assess the situation. Review the raw data and procedure. Check whether the correct standard and equipment were used. Verify the calibration status of reference instruments. Check for leaks, bubbles, contamination, incorrect settings, or other obvious causes. If required, initiate the applicable device deviation out of calibration or OOC or investigation procedure according to laboratory quality system. After corrective action, repeat testing only according to the approved procedure. Most important, the original failed result must not be hidden or discarded. Data integrity. Data integrity is a critical part of HPLC calibration. All calibration data should be attributable, legible, contemporaneous, original, or a true copy and accurate, together with the applicable complete data integrity expectations. Electronic records should be maintained in the validated chromatographic data system or other approved system. Any changes, reprocessing, or modification should be appropriately controlled and documented. Never manipulate raw data simply to achieve a passing calibration results. Calibration documentation. Complete documentation is essential. A calibration record should normally contain information such as instrument identification number, calibration date, applicable SOP, reference standard identification, reference equipment identification, raw observations, calculation, acceptance criteria, final results, and required approvals. The exact documentation requirements depend on the laboratory quality system. Calibration level. After successful calibration, the instrument status should be updated according to the laboratory procedure. Where applicable, the calibration level may identify the calibration status and due date. If an instrument fails calibration or its status cannot be confirmed, it should not be used for routine analysis unless specifically permitted under an approved procedure. Common mistakes. Let's discuss some common mistakes. Number 1. Using an expired or unsuitable reference standard. Number 2. Using a measuring device whose calibration has expired. Number 3. Starting measurement before adequate stabilization. Number 4. Using incorrect instrument setting. Number 5. Recording observation later instead of contemporaneously. Number 6. Ignoring a failed result. Number 7. Repeating a test simply until a passing value is obtained without following the investigation procedure. And number 8. Incomplete documentation. These practices can seriously affect confidence in the calibration record. Calibration frequency. How frequently should an HPLC be calibrated? There is no single frequency that applies to every laboratory and every HPLC system. The frequency should be stabilized through the laboratory approved calibration program, manufacturer recommendation, applicable requirements, equipment, history, risk assessment, and intended use. Many laboratories are predefined periodic calibration intervals, but the actual frequency must be determined by the applicable quality system. Always follow your approved SOP. Calibration and qualification life cycle. HPLC calibration is part of a larger equipment life cycle. The instrument may go through design and installation qualification IQ, operation qualification OQ, performance qualification PQ, routine operation, preventive maintenance PM, calibration, periodic review, and eventually retirement. Calibration should therefore not be considered an isolated activity. It is one part of maintaining the controlled state of equipment. Practical analyst tips. Here are some practical tips for analysts. Always read the current approved SOP before starting. Check the calibration status of every reference instrument. Verify standard identity and validity. Allow adequate system stabilization. Record raw observation immediately. Do not round value before performing calculation unless the procedure specifies rounding. Do not change acceptance criteria based on the observed result. Do not hide failed results and always maintain complete traceability from the reference standard to the final calibration report. Final summary. Let's summarize. HPLC calibration is performed to demonstrate that important instrument performance characteristics remain within predefined requirements. Depending on the system and approved procedure, calibration may include checks such as flow rate, wavelength accuracy, temperature, injection performance, gradient proportioning or GPB, and other applicable parameters. The key principles are: use appropriate and valid reference standard, use calibrated measuring equipment, follow the approved procedure, record raw data accurately and contemporaneously, compare result against predefined acceptance criteria, investigate failures appropriately, and maintain complete data integrity and documentation. Most importantly, never assume that one calibration procedure or acceptance criteria applies to every HPLC. Always follow the specific approved SOP, manufacturer requirements, and applicable regulatory or pharmacopoeial requirements. Remember, reliable instrument performance support reliable analytical data and reliable analytical data supports reliable pharmaceutical quality decisions. I hope this detailed explanation of HPLC calibration was useful for you. If you work in pharmaceutical quality control, analytical method validation, stability, or laboratory operation, understanding instrument calibration is an essential part of good laboratory practice. Thank you for watching.